Hello, student friends. Myself, Dr. Vikrant Pawar, Assistant Professor, Department of Animal Genetics and Breeding, Mumbai Veterinary College. Today, we are going to deal with the problems based upon the modification of dihybrid cross. So, before starting with this particular practical class, let's have the recap. Everybody knows that Gregor John Mendel, he has given the two basic laws of inheritance. First was laws of segregation, and second one was the law of independent assortment. Within the first law, Mendel has crossed the tall plant, homozygous tall plant with the dwarf plant. And in F1, he has obtained that all plants were tall plants. And when these F1s were cell, he has strikingly has got the results like that. There were the tall plants as well as the dwarf plant, which were not, dwarf plants were not seen in F1 cross, but they have reappeared. That's why he has stated that these members of pairs of contrasting character, that is these genes, they get segregated out during the gamete formation and they remain unblemished within their crosses. That means dwarf, whatever the genes or alleles responsible for the dwarfness or tallness, they will not mix together, they maintain their identity. And within that, he has considered only one characteristic, that is the height of the plant, which are governed by the only one pair of alleles. So that's why it is considered as a monohybrid cross. And in F2 ratio, he has obtained the phenotypic ratio, that three tall plants versus one dwarf plant, that is 3:1. And in genotypic ratio was 1:2:1, that is one was the homozygous tall plant, two were the heterozygous tall plants, and last one was the homozygous dwarf plant. So in this way, he has given the first law of segregation. That is, in second law, he has considered two plants simultaneously. Two plants simultaneously. In that particular cross, he has considered two characters simultaneously, that was the color of the seeds as well as the shape of the pod. So in this particular cross, he has considered the two characters at a simultaneous. So that's why it has been said as a dihybrid cross. So cross between the two parents that differ by two pairs of allele. That is, here you can see that there is a pair of allele, that is capital A, capital BB. Whenever it is crossed with the small a, small b, when these two crosses were done, he has characterized, or he has considered the two different characteristics, like the shape of the pod as well as the shape of the color of the pod. Similarly, he has considered the color of the seeds as well as the softness of the seeds, whether it is a wrinkled or a round one. So on the basis of these crosses, he has given the second law of, that is the law of independent assortment, which states that members of different pairs of genes segregate independently of one another during gamete formation and recombine in every possible combination in subsequent generation. So whatever may be the two, here the two pairs of alleles are there, AA and BB, while the small a and BB. Whatever may be their parental combination, these alleles, two pairs of alleles, they get asserted independently. That means here capital E is paired with capital B. Here in small a is paired with small b. But whenever there is a gamete formation, the A can pair with B, or A can go for the small b. Or similarly, capital B can go with A, or it can remain. So it can assert independently, and it can combine in all possible ways. So that's why for the proof, he has considered, he has given one experiment, or he has conducted one experiment where he has cross pure breeding round yellow seeds variety of the sweet pea plant or garden pea plant, which was crossed with the pure breeding wrinkle green seeds. In F1, he has got all round yellow seeds. When this F1, that is having the all round yellow seeds plants, were crossed with the F1s having the all round yellow seeds, in F2, he has strikingly has got 4 different types of the phenotypes. The round yellow plants with the round green seeds, brown plants with the wrinkled and yellow seeds, while the wrinkle and green, in a proportion of 9:3:3:1. So this was considered as a Mendel's classical dihybrid ratio, because he has considered two characteristics or two pairs of alleles simultaneously. Now you can see here, like if you consider the genotype of the yellow and round seed, that was capital YY, capital RR, and wrinkled and green seeds, where it was the YY small RR. So when these gametes are formed, the F1 were the having the genotype capital Y small y, capital R small r. So that means here yellow is dominant over the green, while the round shape is dominant over the wrinkled one. So this all F1s, everybody knows that they were yellow and round. So when these F1s were self, when they are crossed among themselves, instead of staying whatever the parental gametes, see like here the parental gametes were capital Y and capital R, while small y and small r, instead of staying in the same combination, this capital Y not only combine with the capital R like this, similarly capital Y can combine with the small r. Similarly, capital R went with the capital Y, as well as the capital R went with the small y. So in this way, there is an independent assortment, and whenever these F1s or their gametes were crossed or mated amongst themselves, it has produced 16 different types of the combinations in a proportionate that if you compare, there were the 9 out of 16, there were 9 were yellow round, out of that 3 were green round, 3 were the yellow wrinkled, and 1 by 16, that is a last one, that is homozygous recessive for both pairs, it was a green wrinkled. So in this way, the Mendel has covered or he has discovered the law of independent assortment, which has given the classical Mendelian ratio of dihybrid cross, that is 9:3:3:1, that is the F2 ratio. So this was the normal phenomena, but Mendelian laws, that is law of inheritance, they are not universal, because there were so many modifications they are there, because first thing, these modifications are due to because Mendel has considered only one kind of the gene action, that is a dominant gene action. Also, whatever the characteristic he has selected, they were fortunately located on the different pairs of chromosomes, as well as they are clear-cut distinguished contrasting characters. And last one, there is no interactions between the different genes. So that's why whenever the different scientists, they have studied the different laws of inheritance, they have discovered that Mendelian genetics is not a universal one. There are the certain modification. So based upon this, let's see that what are the modifications of this particular dihybrid cross ratio. Everybody knows that we have the classical dihybrid phenotypic ratio is 9:3:3:1. So, but whenever it was based upon the complete dominance, but there can be, when there are two pairs of they are combining together, out of that one pair may be having the complete dominance and another pair or both pair might be having the incomplete dominance or lack of dominance, where in such cases your expected phenotypic ratio, F2 ratio, can be modified. Like let's see here in the first case, where the first locus is either dominant and recessive, where the second locus pair, that is second pair, it is a co-dominant. In that case, the expected phenotypic ratio is 3:6:3, 1:2:1. While whenever both pairs, they are co-dominant, in that case, we can expect the ratio 1:2:1, 2:4:2, 1:2:1. So here, instead of 4 phenotypes, we have the 6 different phenotypes, while here we have the 9 different phenotypes with the altered dihybrid ratio or classical ratio. Similarly, there is a gene interaction. First one was that there is a lack of dominance or there is a co-dominant gene action. Sometimes there is some 2 genes or 2 pairs of alleles, they interact with each other to produce a different kind of the phenotypes. Like here, chicken comb pattern in case of the poultry, where the classical dihybrid ratio was 9:3:3:1. Like whenever there is a pea comb variety was crossed with the rose, F1 were the walnut, and when these walnuts were crossed amongst them, it has produced 4 different types of the phenotypes. Initially, there were the as in proportion of 9 walnut, 3 rose, 3 pea, and 1 was the single comb variety, which was neither in F1 cross or neither in a parental generation. So that's why, though it is the ratio, it remains 9:3:1, but we have the additional 1 phenotype. So this is the first kind of the modification of the Mendel's dihybrid ratio. After that, there is a in previous cases, there was a gene interactions or intra-allelic gene interaction where the, in case of other, there is a term called as epistasis where the, there is a masking effect of one pair of allele with its non-pairing pair of allele. Based upon that, the epistasis, there is a, this Mendelian dihybrid ratio changes. Like here, we can see that classical ratio is 9:3:1. Now, in second case, there is called as a dominant epistasis, whereas there is either of, like you can see here, capital A, capital B, or these are the allelic or you can say genotypic combinations. Both are capital. One is capital, another pair is recessive. Alternate, first pair is recessive, another is the capital allele, while the last one is both are homozygous recessive. So whenever there is a either of one allele is a dominant over the its non-allelic pair, it results into the dominant epistasis where the your classical ratio is changed to the 12:3:1. Okay. Similarly, recessive epistasis, it stands for the either one of the allele which is in a homozygous recessive condition, it masks the expression of its non-allelic pair. So where it results into the 9:3:4, where here small a is able to mask the expression of the capital B, what irrespective of it is dominant over its own allele. So that's why ratio is altered. Here we have only 3 phenotypes, 9:3:4 in proportion. So there is an incomplete duplicate recessive gene interaction. So where there is, it is similar to the that of the duplicate recessive epistasis, where there is a out of 16 combinations or for 15 combination, it acts as a whenever there is a either of the one allele, that is small a or small bb, that is in homozygous recessive condition, it will mask the expression of its non-allelic pair, wherein whenever there is a small a, small b, that is both allele, both pair of allele, they are in homozygous recessive condition, it will be a another characteristics or another phenotype. It is due to the interaction. So incomplete duplicate recessive epistasis with the interaction, that's it changes. The ratio is 9:6:1. Similarly, we have the duplicate dominant epistasis. Duplicate dominance, as name indicates, there is a dominance at both alleles. That is either A, if A is dominant, then it will mask the expression of its all non-allelic pairs. It will mask the expression of its own allele as well as non-allelic pairs. Similarly, so that's why, and is here the name indicates duplicate. So either if A is dominant or B is dominant, it will mask the expression of its non-allelic pair. So here you will have the ratio, phenotypic ratio, only 2 phenotypes will be seen. That is whenever there is a dominant, like both alleles, both non-allelic alleles are dominant, in that case it will be the 15, while only the homozygous recessive in both pair of allele, it will be another phenotype. So here the 15:1. Similarly, duplicate recessive epistasis, as name indicates, duplicate recessive. So here in either of the any allele, that is either small a or small bb, it will mask the expression and your phenotypic ratio will be altered from 9:3:1 to the 9:7. Similarly, where there is a seventh is the dominant recessive interactions or epistasis, where one pair is dominant, where the capital A is dominant, it will mask the expression of another allele. Similarly, if another its alternative non-allelic pair, if it is in homozygous recessive condition, it will mask the expression of it another allele, even it is there is a dominance. So here this classical ratio is modified as a 13:3. So these are the some background related with our practical class. That's the not only the Mendel has discovered the law of independent assortment. He has stated that the classical ratio as a 9:3:1, but there are the certain conditions where either there is a lack of dominance in one pair or lack of dominance in both pair, or there may be gene interactions, or there may be the epistasis where the non-allelic pairs interact with each other, where the one pair can mask the expression of its non-allelic pair, and which results into different phenotypes. Now let's come to our today's practical, that is problems based upon the modification of dihybrid cross ratios. Now let's see the first example. Here these are the example. There are the two pairs of alleles govern the color in mouse. Crosses were made between the black and albino mice. The F1 mice were all black. In F2, the mice colors were as follows: 92 black, 30 cream, and 49 albinos. Find out the what proportion of offsprings would be black, cream, and albino. Give the necessary explanation. Now, coming back to this particular problem. What is given? It has been said that the color of the mouse is governed by two pairs of allele. Definitely, that means it is governed by two pairs of allele. It is a dihybrid cross. Okay, that is clear for us. Now, second thing, they have given there are original two colors were there, black and albino mice were there. Crosses were made, and after that, in F1 were crossed. The F2 were 92 black, 30 cream, and 49 albinos. So black mice were in 92, cream 30, and albino were 49. So in total, there were 171 mice were there. Okay. Now, first we have to, for identification of whether what is the proportion of offspring that would be black, cream, or albino, we have to find out its ratio or proportion of each phenotype with respect to the dihybrid cross. So we know that the classical dihybrid ratio is 9:3:1, and total 16 combinations were there. So how to find out proportion of each phenotype in this particular example? So what we have to do is, like, say for example, black, we have original 92 mice were of a black color, 30 were cream, and albinos were 49. Now let's talk about the black mice. So there were 92 mice out of 171. That means the proportion would be 92 of 171. Now we have the 16 combinations. So to find out the proportion, we have to find out first the proportion of these black mice to the total mice, and it has to be multiplied by the 16. That is 16 combinations we are, different genotypes we are expecting. So 92 divided by 171, that is 92 is a number of black mice or this particular phenotype, divided by total number of mice multiplied by 16. So that ratio comes as, or this calculation comes as a 8.608. Similarly, for cream, again the same procedure, 30 divided by 171 multiplied by 16. The proportion is, or that answer is 2.807. Now for the third one, again 49 divided by 171 multiplied by 16. That means 4.584. So if we look at these proportions of each phenotype, that is black, cream, and albinos, so their proportions would be 8.608, means it is almost 9, approximately 9. Here 2.8 means it is approximately 3, and here 4.5, a little bit larger, but it is approximately 4. So you can see that these are very close to the 9:3:4 ratio. That means ratio between the, or proportion between the black, cream, and albino, that is 9:3:4 ratio. That means we know that we have seen that 9:3:4 ratio means it is a recessive example of recessive epistasis. Once again, just I will explain. To find out any proportion of each phenotype to the total with respect to the dihybrid cross, we have to take a ratio of the individual number of phenotypes to the total phenotypes multiplied by the 16. So in this way, we have to find out proportion of each and every phenotype that we have. So after that finding, we have to compare, and we have to try to compare with the modified ratio. So after comparing, we know that 9:3:4 is a classical recessive epistasis example. So let's see, try to explain how it can possible, how we can get a 9:3:1 with a specifically actually by considering the genotype as well as its crosses. Now, when the black mouse, homozygous black mouse, which has the it is governed by two pairs of allele, it is already given. Let's consider as the capital RR, capital CC is the genotype of the homozygous black mouse, which is crossed with the albino mouse. Albino mouse, it is again, it is small RR, small CC. Okay. So when it is crossed, the F1 would be the capital R, small r, capital C, and small c. Since they both dominants are coming together, all F1s would be black. Since capital R and capital C, there is a combination, it will be that all offsprings will be black mice. Now, when these F1s were crossed among themselves, that is that black mice with F1 black mice with the F1 black mice, definitely we know that their genotype is capital R, capital C, small c, cross with the capital R, capital C, small c. Now, we know that due to the independent assortment, these alleles, whatever the non-allelic pairs, they will get segregated out in a parental combinations. They will assort independently during the gamete formation. So here we will have 4 different types of the gametes: capital R, capital C, capital R, small c, similarly small r, capital C, and small r, small c. So in this way, we have 4 different types of the gametes. So if you do the like Punnett square method, so where we have, if you go for the crossing, there will be the male gametes, 4 gametes would be there, female 4 gametes would be there. So after crossing, like after having these particular crosses, what would be the genotype and phenotype? Let's go here. So here, capital R, capital C, capital C. So if you look at all the black boxes that I have with the black phenotype, you will find that there is at least one allele of each pair will be dominant ones, capital R and capital C. Whenever this combination is there, capital R and capital C, the phenotype will be a black one. Now, in this case, why it is happening? You have to look at that some background, because here the capital R is responsible for the black, but it can produce its black pigment when there is another pair of the capital C is there, that another pair of allele which allows for the pigmentation, So whenever there is a capital R and capital CC, or any like capital R and capital C, it will be always a black. But now you can look at here. Now, whenever we have the capital C, it will allow for the pigmentation. But it is when it is in the small cc, that is homozygous recessive condition. That homozygous cc, it will not allow for any pigmentation. So whenever we have the this homozygous cc combination, that is small c, even if that capital R is responsible for the black pigmentation, it will not be expressed. That's why it is a albino. Similarly, if we can find out which are the combinations which are having the small c. So this is the 1, 2, 3, and 4. So here we have the 4 albinos because we have the small c. That's why it is called as a recessive epistasis. In a homozygous recessive condition, that is small c, it will mask the expression of its non-allelic pair, that is capital R, or even it is small r. So this small r, it is only expressed when there is a capital C is there. Okay. So here, whenever we have the homozygous small r, capital C or small c, that 3 phenotypes would be the cream color. So in this way, we have proved that first one, we have calculated the phenotypic ratios, and on the basis of that phenotypic ratios, we have tried to justify or proportion of each phenotype, and with the help of this previous knowledge, we have tried to find out how this particular example is of a recessive epistasis, where we have got a 1, 2, 3, 4, 5, 6, 7, 8, and 9 black. For production of the cream color, which one should have the small rr with 1 capital C. So when these are the small rr with capital C, 3 combinations are there. And last one, there are 4 albinos were there, which are result of small cc, which do not allow for production of any pigment. So in this way, we have discussed the first example, that is the recessive epistasis. Now, coming back to the next example, where the 2 yellow center daisy flowers strains were cross producing all F1 with the only purple central flowers. So what is given? There were daisy flowers which comes in a 2 different yellow centered colors, like one with the yellow flowers with the yellow center and yellow flowers with the purple center. Now, 2 yellow center varieties they were crossed. Random crossing amongst them, F1 produced 96 progeny plants. They have crossed what? the purple varieties. Initially, they have crossed the two yellow center variety, daisy flowers, which has producing only the purple flowers. So now these purple flowers were crossed randomly, and they have found that the F1 produced 96 total progeny plants, out of which 53 were purple center flowers and 43 were yellow center flowers. So let's see what will be the phenotypic ratio of F2 generation and what type of gene interaction involved, and we have to explain in detail. So what is given here? Now let's go to back. Now they have clear cut say that two daisy flowers are with the yellow center variety, they were crossed. That is the first thing. Now when these two yellow center varieties they are crossed, they are producing a plants with the purple centered flowers. That means first thing, it indicates that this whatever the two yellow flowers, they are not genetically identical, they are genetically different. That is the first one. Second things, whenever the purple center varieties they were crossed, we can expect that 9:3:3:1 ratio as in case of the gene interaction. But again, it has only two phenotypes with a proportion. So first we have to find out the proportion as we did in the last example. So let's see that purple center varieties were 53, yellow center varieties were 43. In total, there were 96 flowers were there. So we have to find out the proportion with respect to the dihybrid cross. So 53 by 96 as we have done in the previous one. 53 purple divided by total number of flowers, we multiply by 16. So ratio comes 8.83, which approximately 9. Again, yellow center varieties 43 divided by 96 multiplied by 16, which is a 7.16, which is approximately 9:7. So proportion or ratio between purple to the yellow, or proportion between purple to yellow, is 9:7 ratio. So if we can recollect 9:7 ratio, that means it is a duplicate recessive epistasis. It is an example of duplicate recessive epistasis. Now we have to prove with the help of the suitable example. Now I have given here the pictorial diagram like duplicate recessive epistasis. Here what we have considered that daisy flowers with yellow flowers with purple center. This is the one example. Daisy flower with the yellow center, yellow flower with yellow center, and yellow flower with purple center. These are two varieties where. So in our example, it has been given like two yellow center varieties were crossed, yellow center with yellow center. It has produced a purple center. Okay, but F2 ratios were 9:7, 9 purple as 7 yellow center flowers. That means it is a duplicate, it is a recessive epistasis. Duplicate recessive epistasis indicating two varieties are genetically different. So let's say actual. So as since it is a duplicate recessive epistasis, yellow center variety will be yellow whenever there is either small pp will be there or small rr will be there. Since we have crossed two different yellow center varieties, because it is producing a different purple center class, so their phenotype would be capital PP small rr with another yellow center variety is a small pp with capital RR. So look at the genotypes here. So since it is a duplicate recessive epistasis, whenever there is either small pp or small rr is there, it will be a yellow center variety. But whenever it is cross, this capital PP small rr, whenever it is crossed with the small pp capital R, F1 will be the capital P small p capital R small r. That means it is a heterozygous for both pair of allele. Okay, so since there are one capital P and one capital R, they are coming together, they will interact in such a way that it will produce a purple. Since there is no here in this particular combination, there is no pair with a homozygous recessive. These are the heterozygous. So in that case, the F1 are producing a purple. So whenever we have the one capital P and one capital R in any genotypic combinations is there, that phenotype will be a purple. That is one has to understand and keep in mind. So whenever these purple color varieties they were self or they are cross amongst themselves, it can produce a, as we know that it can produce a 4 types of the gametes: capital P capital R, capital P small r, small p capital R, or small r small r and small p. So these 4 different types of the gametes can be produced. So now why it is happening, why we are getting the yellow center 9:7 purple versus 7 yellow center daisy plants? Because whenever the genotype is having at least one dominant allele of each pair, that is capital P and capital R, it would be a purple. Whenever we have either small pp or small rr is there, irrespective of whatever the another allele is there, whether it is a capital R or capital R, small r, or here in case of the whether it is capital P or small p, whenever there is a small rr, it would be a yellow center variety. That's why it is called as a duplicate recessive epistasis, because on either pair of allele, whenever it is homozygous recessive, it has a epistatic effect where it's non-allelic pair, and it will mask the expression of its non-allelic pair. So that's why here we got a 9 purple to the 7 yellow. The same thing has been depicted here, like in a crosses we have discussed, like whenever there is a at least one capital P and one capital R, it would be yellow. Only genotypes or only the crosses where we have the small rr or small pp, either small rrs or small pp, it would be a yellow. So here this is a small r, because of this small r it is yellow. Here again it is because of small r it is yellow. In this case there is a small pp coming together with a capital R. Even there is a capital R, you can see that it is a yellow. Similarly, this is a yellow due to the small pp. Here it is yellow due to the small rr, and here it is due to the small pp, and here both pair of allele is homozygous recessive. That's why it is a yellow. So in total there are the 9 purple versus 7 yellow. Hope you understood the basic concept. Since it is a duplicate recessive epistasis, any one of the pair of allele, if it is homozygous recessive, it will mask the expression of its non-allelic pair, and that's why, and both pairs having the recessive epistasis, it is called as a duplicate recessive epistasis. Okay, now let's move to the third example. The black Langshan breed of the chicken has a feathered shanks. When these birds are crossed with the buffalox breed with the unfeathered shanks, all F1 progeny have the feathered shanks. Out of 360 F2 progenies, 24 were found to have non-feathered shanks, while remaining 336 had feathered shanks. What is the mode of interaction, and what proportion of the feather in F2 generations are expected? Okay, now again, here there are 2 different breeds of the chicken. They are or poultry. They are given black Langshan, which is the characteristic of the completely feathered shanks, while the buffalox, they are the clean shanks, means they don't have the feathered. So whenever these birds were caught, the breeds were like black feathered shanks were crossed with the clean shanks or unfeathered shanks of buffalox. The F1 progenies were feathered shanks, and when these feathered shanks they were mated among themselves, F2 were predominantly there had been a feathered shanks. So let's see what is the proportion of each feather, feathered versus non-feathered shanks. So here the feathered shanks are 336, non-feathered shanks are 24. In total, there are 360 birds with the feathered as well as non-feathered shanks. So proportion of the bird with respect to dihybrid cross, again the same procedure. We have to take a ratio or proportion of feathered shank to the total birds, multiply by the 16. So it is a 14.93. Similarly, non-feathered shanks, we have the 24 by 360, multiply by 16, it is a 1.06. So in approximately first is a 15:1 ratio. So 15:1 ratio, we know that it is a duplicate dominant epistasis. It's a case of duplicate dominant epistasis. Means either any one of the allele, either it is, suppose it is governed by the 2 pairs of allele. Let's say, for example, feathered, we have the capital FF feathered shanks, and we have another alternate pair of allele which is small SS, which is for the which is representing the non-feathered or clean shanks. Okay, so capital F and small f and capital S and small s, it's a pair of acting on the shank feathering. So here, since it is a duplicate dominant epistasis, means at both alleles, whenever they are in a dominant in nature, they will mask the expression of its own allele as well as non-allelic pair. So even in every G, if even in H genotype, if you have the either capital F or capital S out of 4 alleles, if it is a capital F or capital S, all would be the feathered shanks. Only bird that would be the clean shank would be the small FF, small SS, because there is a neither capital F is dominant or nor the S is dominant. Okay, which is responsible for the clean shanks. So in this case, only the small FF, small SS, that is 16th cross, last cross would be the clean shanks. So parental crosses are there. Let's see, consider that feather shank, that is homozygous. It is a capital FF, capital SS. When it is crossed with the unfeathered shank, as everybody knows that it would be its genotype is only small FF, small SS. So all F1 would be the capital F, small f, capital S, small s. All F1 would be the feathered shanks. Whenever the F1 are crossed amongst themselves, feathered with feathered shanks, it will have the 4 gametes, male gametes, capital F, capital S, capital F, small f, capital S, and small s, F, small s. So both male and female, we have the all 4 types of the gametes. So whatever the crosses with genotype with the capital S, capital F and capital S, capital F with the small ss or small ff with the capital S, whenever either capital F or capital S is a dominant in nature, it will be a feathered shank. So that's why it is called as a duplicate dominant epistasis. Either capital F or F is dominant or S is dominant, that allele, it will mask the expression, and all that bird would be feathered shank. Only bird that would be a unfeathered shank or clean shank is a having genotype small ff, small ss. So F2 results are 15 feathers versus 1 unfeathered shanks. So you can look at here. So these are the crosses, 16 crosses are there. Only one 16th combination or 16 genotype, it has a both alleles as a homozygous recessive. Otherwise, each and every cross, either he does one capital S is there, or here it may be the only one capital F is there. Even if is there, it would be a feathered one. So this is the example of duplicate dominant epistasis, where it is governed by the both pair of allele. When they are dominant in nature, they will mask the expression of its non-allelic pair, and the 9:3:3:1 classical ratio is altered into the only 2 phenotypes as a 15:1 proportion. So this is the third example. Now let's see the last example based upon the dihybrid ratio, is that whenever the red color Duroc Jersey pigs is produced by the genotype capital RR, capital SS. So there are the Duroc Jersey breeds, they have the red skin coat color, while there is another white coat color is due to the double recessive genotype, that is small r, small s, small s. The genotypes of capital RR, small ss, and small rr, capital SS produces sandy color. A homozygous red variety is crossed with a white variety. What the phenotypic results are expected in F1 and F2 generations. Now, if you just look at the back to this particular example. Now there is a Duroc Jersey breed. It has a 3 different types of the skin coat color as it is given. Like Duroc Jersey red color is due to the capital R, capital S, capital S. That means at least one allele of each pair of allele, it should be a dominant. It will in combination capital R and capital S, it would be a red color, while both pairs of homozygous recessive, that is small r, small s, in that case small rr, small ss, that would be that phenotype will be white. But whenever there is a one pair is dominant and alternate pair is recessive, that is capital RR, small ss, or small rr, capital SS, it produces a third type of the color that is called as a sandy color. Okay, so here we have been given that is actually crosses. So we have to find out. So whenever this red variety with the capital RR, capital SS, which is crossed with the small rr SS, what would be the F1 and F2 phenotype, and what is the proportion? So let's see the phenotype of and genotype of the each Duroc Jersey pigs. Now red color, it is expected that as it is given, it is a homozygous. It is given as a homozygous capital RR, capital SS, that is, it is a red color. Or if we have the one capital R and one capital S, as that first example we have seen a purple color or like that, So when we have the two both alleles which are out of that one of the allele, that is capital R and capital S, if they are dominant in nature, it will be a red color. Now sandy color is there. It's look like as a duplicate recessive epistasis, like whenever there is small ss or small rr, whatever may be the capital R or capital SS, irrespective of that capital R here due to the small ss, or here due to the capital SS, that is capital S or homozygous dominant S, it is a sandy color. So it looks like a duplicate recessive epistasis. But there is a twist, that because the 16th, that is last cross, last combination, that is, it is a homozygous recessive. Both alleles are homozygous recessive. In that case, it is producing a third different types of the color. So here it is a, you can see that there are three phenotypes instead of, so it is neither completely duplicate recessive interactions, but there is something involved, there is a gene interaction also. So you can say it is a duplicate recessive epistasis with a gene interactions. So let's see what are the parental crosses. Red, as we have discussed, capital R, capital SS. White with the small r, small s. Red would be the F1 would be the red. When the F1s are crossed, again it will produce a male, both four types of male and female gametes, capital R, capital S, capital R, small s, small r, capital S, and small r, small s. So here, now what are the phenotypes based upon the genotypic combinations? Whenever we have the capital R and capital S, it would be a red. When we have the small r, capital S, or whenever we have the one capital R with the small ss. So here it is one of the important point to be noted, that this homozygous recessive pair should be accompanied with another dominant allele. Either one allele should be a dominant. Means here the small r with a one capital S, it is sandy color. Where it is a one capital R with a small ss, it is a sandy color. So, but whenever both alleles are homozygous recessive, small r's, it is a white color due to the gene interaction. So here we can expect that is a 9. So 9 red, 6 sandy, and 1 white. So it is a duplicate recessive epistasis with a gene interaction. Or you can incomplete duplicate recessive epistasis with a gene interaction is the example. Here we can see by actually crossing, as 4 gametes of male, female gametes, they are crossed. We have the 16 combinations. So whenever we have the one capital R and capital S, it would be a red color, that both combinations are there. There are 9 crosses would be there of a red color. Whenever we have the one dominant allele and its respective non-allelic pair is a recessive, it would be a sandy. That is here capital R, small ss. Here also one capital R with a small ss. Here again here capital R with small ss. Here 3 combinations. Similarly here we have the small r with a one capital S. So again 3 combinations are there where we have the small r with a capital S. At least one is a dominant one. In that case, we are getting sandy. So there are in total 6 sandy and only 1 is a white. So we are getting 9:6:1, indicating it is a incomplete duplicate recessive epistasis with a gene interactions. So today we have discussed in this particular lecture or a practical class that the Mendelian Dihybrid ratio, that is 9:3:3:1, is a universally accepted as a classical Mendelian ratio, but there would be due to the gene interactions, or there will be the lack of dominance, or there is a co-dominance. Because of that, whatever it is not always holds true that there would be always a that you will get a classical 9:3:3:1, that is 4 phenotypes in a F2 ratio. So here we have seen the few different examples, how we can calculate their respective proportion, and how we can compare and how we can demonstrate the different modifications of the dihybrid ratio. First, we have discussed with the recessive epistasis. Second, we have discussed duplicate recessive epistasis. Third one is a duplicate dominant recessive epistasis, and last one is the we have discussed the incomplete duplicate recessive epistasis with the gene interaction. So here we will stop. Thank you for the patience listening, and you can refer this video for further studies. Thank you.